drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a Ph.D. in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. This episode is ad-free, and I rely on my audience to support my efforts. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode.
Yeah, thank you for having me on once more.
Can you just remind everyone, especially those that did not already listen to our first conversation from last year, who you are and what your lab studies and what you guys are interested in?
Yeah, so I run a lab at the University of Copenhagen in Denmark at the so-called Nordisk Foundation Center for Basic Metabolic Research. And we can already kind of try and tackle the name right now, because I think a lot of people associate our research center with the Novo Nordisk, the company, but it's different. It's a foundation that is related to the company, but they donate money to the University of Copenhagen that then established this research center. So it's one out of several Novo Nordisk Foundation-funded research centers at the University of Copenhagen, and our center focuses on basic metabolic research. But in my lab, we're not only interested in the basic metabolic research, but also in translational metabolic research, and by that I mean trying to take the knowledge that we kind of decipher and learn and develop new pharmacotherapies for treatment of metabolic disease. Most notably, we're interested in obesity. So we're doing a lot of kind of preclinical work with new potential weight loss medications. So I've been running a research lab in Copenhagen since 2017. Before that, I spent quite a few years in Germany, in Munich, and I've also been in the States in Cincinnati working during my Ph.D. studies. And my lab is currently, I think, comprised of 14 or so individuals, a mixture between bachelor and master students, Ph.D. candidates, and postdocs. And next to that, I have a small biotech company where we also try and further develop or commercialize some of the things that we work on in the academic lab, in collaboration with the University of Copenhagen and the kind of this technology transfer unit at the university. So a lot of stuff going on.
In your view, what causes obesity? Is it all about energy balance? Is it exposure to contaminants in the environment? What are the major drivers of obesity?
It's a tough opening question. I think the safest short answer is we have no clue right now. I think that's kind of interesting if you look at also some of the kind of summarizing review work that is coming out these years. I think researchers are more and more open to newer theories as opposed to just 5-10 years ago, where it was kind of oftentimes more clearly stated that the modern environment with its readily available, highly palatable, energy-dense, cheap food is the primary driver of the modern obesity epidemic. It must be, right? But then there's evidence coming in kind of suggesting that it may be more complicated than that, which includes, I mean, and there's a variety of different work here. And you've also had, I think, John Speakman on your podcast, who has done work on basal metabolic rate using doubly labeled water over decades to show that there seems to have been a quite weird but interesting subtle decline in basal metabolic rate over the last 30 years or so, but actually no decrease in energy expenditure from physical activity, which is a little bit counterintuitive what we oftentimes say, like that it's the decrease in physical activity. And at the same time, they've done work showing that there's no obvious correlation between density of fast food, full service restaurants, and obesity numbers. So there's kind of these pervasive ideas that it's the fast food and it's lack of physical activity that is driving the epidemic that is not necessarily 100% supported by evidence. So I think, I mean, we must acknowledge it's something in the modern environment, but it's not necessarily, I think, exclusively something to the food environment, but a combination of factors that and that's the safest answer, right? Something in the modern environment. So it's the safe kind of gene environment interaction, right? But there's definitely something with the modern environment that is that has very negative consequences in the interaction with our genome that elicits that many of us develop excess body fat mass.
Yeah, I mean,
but narrowing it down further than that is really it's really difficult.
Yeah, I think, from talking to John Speakman and Robert Lustig and a bunch of other people on the podcast and thinking about this, to sort of re-summarize and build on what you just said, the naive assumption or explanation for what causes obesity is, well, we live in the modern world. We have a surplus of highly palatable, cheap foods. People are simply eating more calories and they're moving less, right? So we're lazier and we're more indulgent. Therefore, we're getting bigger. But as you pointed out, and as others have pointed out in different ways, that can't be the full picture, because actually we're not moving less than we were a few years ago. We're actually moving a little bit more, and our metabolic rate is going down, and no one really knows why. And we're actually not, we're not all eating more than we were 5, 10, 15, 20 years ago. So the eating more, moving less hypothesis can't be the explanation. Certainly the amount of calories we eat, the types of calories we eat, and other things in our environment have to feed into the explanation for why obesity is moving in the direction that it has been. But the simplistic answer that most people would think of from a common sense perspective, we're just eating more and moving less, is not the explanation. We know that for sure.
Yeah, and at the same time, it also kind of conflicts with this notion of that body fat mass and body weight is homeostatically regulated, right? So if we trust that we are equipped with these homeostatic systems that ensures a stable body temperature, stable blood pressure, water-salt balance, there are actually also similar mechanisms in play, right, to ensure that we have stable body fat mass and body weight. So most of us, or some of us, go through our lives decades without counting calories or worrying about how much we exercise, but our body weight is remarkably stable, right? So many of us can rely on this kind of on these homeostatic mechanisms to ensure that we match our intake with expenditure. But still, then there are now individuals then that develop obesity. So there's something, what is it that the environment that triggers this subtle imbalance that over time develops to become a quite large expansion of body fat mass? I think that's still a mystery, right?
Yeah, I mean, so as you said, body weight and food intake, these systems in the body, just like other systems in the body, are homeostatically regulated. So we normal animals, most people for much of their lives, don't have to think about this, right? The sort of cartoon model I think the average person has in their head is, oh, well, you're metabolically healthy when you're young, and then at some point you become quote unquote old, and your metabolism quote unquote slows down, and then you start gaining weight. And that does in fact happen for a lot of people, but A, it doesn't happen for everyone. B, it happens for different people at different times. C, it's starting to happen even for children more and more. So it's not like you just get old and magically you're supposed to just start gaining weight at some point. It's that these homeostatic mechanisms are becoming broken at some point due to X, Y, and Z, and we don't know exactly what is breaking them or what causes them to break when you're 40 years old or 5 years old.
No, and then adding to the complexity is something that we oftentimes put in a different box at biology, and that's the socioeconomic status. So if there is one predictor, if I were to ask people about one thing in order to predict their body weight or BMI, I could only ask one question. I would ask about their socioeconomic status. Basically, I guess that's two questions: that's education and income, and that is by far the best or worst, if you want, predictor for your body weight. So there is again something with our income and education that predicts whether we have a higher risk for developing obesity. And then again, how does that fit with this idea of homeostatic regulation? And then even in the areas of the world where we have the highest prevalence of obesity, you still have individuals, despite they live surrounded by people in their family and their friends have BMI plus 35, they're individuals that are completely lean without worrying about or thinking about their body weight. So they have they're wired differently, right, in terms of homeostatic protection or defense against their weight. So yeah, the complexity is very large.
We're going to spend a lot of time talking today about some recent research that relates to these new and very popular weight loss drugs, these anti-obesity drugs like Ozempic, and these are GLP-1 drugs. And I want to start out by giving people a sense for some of the basic biology around GLP-1 here. So to start, can you just describe what is GLP-1 and what is it doing endogenously in our bodies?
Yeah. So GLP-1 is one out of several hormones that are produced by our gastrointestinal system. So it's produced by a cell type called the L cell in the gut. And typically these hormones, they are peptide hormones, meaning that they're a specific size in terms of they have a specific number of amino acids. They're produced in response to ingestion of meals, right? And they are sensitive with respect to macronutrient and even micronutrient distribution. So we know that historically they were discovered for glucose ingestion. You see a secretion of this glucagon-like peptide 1, GLP-1, and it was then identified that this would affect the beta cell of the pancreas to stimulate insulin release. So the first kind of loop discovered in the middle of the 80s was that glucose ingestion elicits GLP-1 release from the gut that signals in the pancreas to potentiate insulin secretion so we can remove the glucose from the plasma. So it's a part of kind of this glucose regulating endocrine system. And so this was referred to as the incretin effect. So that means if you give the same bolus of glucose orally versus intravenously, you would have a much more pronounced insulin response to the oral load because you get the effect from the incretin hormones. And by the incretin hormones are comprised of the GLP-1 and its close sister GIP. So these hormones were identified in the 70s and 80s, and in addition to GLP-1 and GIP, the most well-known gut hormones are peptide YY, PYY, or there's a hormone called neurotensin, CCK, and a few other more well-known peptide hormones secreted from the gut in some type of mixture in response to ingested meals, right? So the endogenous function is typically referred to as this incretin effect to potentiate insulin secretion so we can remove nutrients from the bloodstream and get it deposited into muscle and fat and liver tissue typically.
So every time we eat, basically a cocktail of different hormones gets secreted, and they're meant to affect different tissues in the body so that our body can effectively utilize whatever it is we've put into it. So if you eat a meal with one nutrient composition and then a meal with a different nutrient composition, that might necessitate higher or lower levels of glucose uptake or other nutrient absorption. And so when everything's working properly, these gut hormones get secreted in some combination that's suited to enable our bodies to uptake what we've actually ingested.
Exactly, yeah.
And so GLP-1 has to get secreted, and then it has to activate GLP-1 receptors. Where I'm assuming that these things are located on many different cell types and tissues throughout the body. What are the major tissue systems that respond to GLP-1 that express the receptor?
Yeah, so initially we've been focusing on the endocrine pancreas that have these islets of endocrine producing cells that produces insulin, the beta cells, or glucagon, the alpha cell, and they are decorated with these GLP-1 receptors. So that's also why this well-known effect on insulin secretion has been early characterized. Then there are also local neurons sitting in the proximity or vicinity of actually where the peptide is produced in the gut. So, for example, a vagal nerve projecting to the brainstem, probably picking up locally produced GLP-1 signals and transmitting it through a nervous signal directly to the brain. And then this GLP-1 receptor is pretty densely expressed in the hypothalamus of the bottom of the, sitting just around the third ventricle of the brain, in the bottom of the brain, and also in the brainstem where the vagal nerves is projecting around these two nuclei called the area postrema and the nucleus of the solitary tract. So specifically in the context of obesity and diabetes, it's the GLP-1 receptors expressed in the hypothalamus and in the brainstem we're interested in, and in the pancreatic beta cells for the insulin secretion. And then it's expressed to a lesser extent in other tissues, and we continuously try to kind of discover kind of smaller or more subtle levels of expression on other, for example, immune type of cells, etc., to try and understand what effect the peptide can have on other cell types and systems. But mostly it's expressed in the beta cells of the pancreas and in these two areas of the brain.
I see. So it makes sense that it's expressed in the pancreas, because if GLP-1 is related to food uptake and nutrient acquisition and the insulin response, it makes perfect sense that you would find it in the insulin, which is where in the pancreas where the insulin is secreted. It's also found in the nervous system. So it sounds like you've got the vagus nerve, which reaches down into the gut, so it can sense the GLP-1 there, relay that information back up to the brainstem. You also have these GLP-1 receptors, you said, in the brainstem and the hypothalamus. How are they sensing that GLP-1? Is it crossing the blood-brain barrier and getting into the brain, or is it getting to those areas in some other way?
Yeah, so this is the intriguing part. So these areas are often referred to as kind of circumventricular organs or sites. So they are kind of the just surrounding the third and the fourth ventricular system. The blood-brain barrier is often referred to as being less fenestrated or more leaky. So, and that system is quite advantageous in the sense that this is where the brain is capable of communicating neuroendocrine in a neuroendocrine fashion with the rest of the body. So, especially in the hypothalamus, the most lowest region, called the arcuate nucleus, is decorated with receptors for circulating hormones, peptides, and proteins, including insulin, but also leptin produced from the adipocytes. So that area of the brain can actually see peptides and proteins in circulation, and is less protected than the rest of the brain. So it's a kind of a neat design in terms of brain-body communication, right? So the brain can pick up on what's happening in terms of nutrient status and the endocrine kind of factors that signal also nutrient status.
I see. So the most of the brain is protected by the blood-brain barrier. So
yeah.
There's tight regulation of what goes into the brain. We don't want bacteria getting in. We don't want toxins getting in. We want to control exactly what's going in, because the brain is sort of the master controller of everything. So we've got this blood-brain barrier that's tightly regulating what gets into the brain. It's preventing a lot of things from getting into the brain. But a couple parts of the brain, special pieces, including the hypothalamus, they are in direct contact with the blood. So the hypothalamus is basically directly sampling what's in the bloodstream.
Exactly. Yeah. So if you do labeling studies in rodents, you put a fluorophore or something on a peptide, GLP-1 peptide, you can see that it up concentrates in those regions of the brain, but it doesn't get further into the brain typically.
So for a neuron in the hypothalamus, say, what happens when GLP-1 binds to its receptor? Does the neuron become more excitable, less excitable? What actually happens to neurons when GLP-1 gets released?
Yeah, that's a good question. And at the neuronal level, you see increased excitability, but it is a G protein coupled receptor, so you don't have this kind of immediate depolarization necessarily with the. So it's perhaps more a sensitization of the neuron in those cells. So one of the neurons that expresses the GLP-1 receptor is called the POMC or the POMC neurons, which is kind of the primary kind of satiating or satiety hormone in the bottom of the hypothalamus. We talk about these yin-yang hormones in a little bit simplified fashion, where the AGRP neurons, and these are because they produce these neuropeptides, express AGRP or POMC. So the AGRP neuron is the hunger promoting neuron, and the POMC neuron is the satiety promoting neuron. And the POMC neuron respond to 1/3 of them expresses the GLP-1 receptor and will then signal or at least sensitize in response to GLP-1 and will have a little bit be excited, have increased excitability.
I see.
And same goes for some of the neurons in the brainstem.
I see. So the neurons in the hypothalamus and elsewhere that have the GLP-1 receptor, they're sensitive to GLP-1. GLP-1 is basically making them more excitable, which may or may not translate into causing those neurons to fire more directly, but will make them more sensitive to their inputs. So you've got these two populations in the hypothalamus in one nucleus. You have AGRP neurons, which promote hunger, basically, and POMC neurons, POMC, which promote satiety, basically. So if the POMC satiety neurons have GLP-1 receptors and GLP-1 goes up, you would expect to promote satiety, more satiation, less hunger.
Exactly, and there are also then a lot of indirect communication. So there's a recent work where they're using a method called fiber photometry. So you can actually in the live mouse, you can read the activity of neurons. Once they fire, you get like a calcium signal that you can pick up on with an optic sensor, a fiber. And then you can see if you give these analogs, GLP-1 hormones, that you also can see a suppression of the AGRP neuron. It's not direct because it doesn't have the receptor. So there's a lot of cross communication going on between these neuronal systems, right? So again, and we can talk about this later also, we see also that activity deeper in the brain with some of these type of gut-based hormones, but it's not because we just talked about, they don't reach deep into the brain, but they will have feed forward or they will have activate neuronal signals that will then kind of signal further into the brain and kind of promote even affect, for example, the mesolimbic hedonic reward pathway. But those are not because the peptide is direct signaling there, but it's through feed forward pathways.
I see. So the hormone GLP-1 can promote satiety at the level of the brain by acting on specific neurons in the hypothalamus.
Exactly. Yeah, and probably in combination with its effects in the brainstem, right? So there's a study from 2018 where they made a GLP-1 receptor knockout animal where they only ablated or knocked out the GLP-1 receptor in glutamatergic neurons. So they used a marker for glutamatergic neurons called the V-GLUT-CRE mouse, and then they crossed it with a GLP-1 receptor flux mouse. So you only knock out the receptor in neurons that expresses this marker for glutamatergic neurons. And then they tested one of the earlier long-acting versions of the GLP-1 drug, and they saw that in mice that didn't have GLP-1 receptors in the glutamatergic system, the drug didn't work on body weight lowering and food intake lowering. So based on that study, for example, you can pinpoint that the pharmacological benefits on body weight and food intake is mediated at least through glutamatergic system in the brain. They did the same for the GABAergic system, and they didn't see any loss of effect of the drugs. So those type of combination of these mouse genetic works with the pharmacology can then teach us more about the central modes of action.
And so because we've got these circumventricular organs that can directly sample the bloodstream of the brain, in parts of that brainstem, the hypothalamus, that means these GLP-1 agonist drugs, the Ozempics and the related drugs, they are actually acting on those areas of the brain directly.
Exactly. And we haven't, this is something we should probably touch upon now because the endogenous hormone is very, very short-lived, less than 2 minutes. So it's debatable whether the endogenous produced GLP-1 ever reaches the brain in a sufficient concentration to signal satiety.
I see.
So we exploit the system with the pharmacology because we can create these super long-lived versions of the hormone that are protected from enzymatic cleavage in the bloodstream. And through lipidation or fatty acid side chain, they also bind to albumin, so they stay in blood circulation for many, many, many days. And in that way, they've been engineered to signal in the brain as opposed to the endogenously produced GLP-1.
I see. So that was actually going to be my next question, is how these GLP-1 drugs differ from endogenous GLP-1. Sounds like one major way is they've been engineered so that they last longer. They're in circulation for longer, and therefore they can promote satiety with a longer time course.
Yeah, and that is the primary difference from the endogenous one. So it's the substitution of a few amino acids to prevent them from being degraded by an enzyme called DPP4. So they don't have this enzymatic breakdown. And then through this lipidation or this fatty acid side chain, that is kind of a well-known modification in medicinal chemistry now, because it then enables your peptide to bind to albumin that circulates in large quantities of the body, and that is then again you retain the peptide longer time in circulation. So this is also why a significant breakthrough in this area by the pharmaceutical companies was from going through the once daily to the once weekly version that is now on the market. So the predecessor to semaglutide was called liraglutide, and it was a once daily GLP-1 receptor agonist, and through further refinement of this side chain, it was made into a once weekly drug, basically.
I see. So through these kinds of modifications, it's a once weekly drug because it's been engineered to basically stay in circulation for longer. The body can't break it down as quickly as it can break down the natural GLP-1 hormone.
Exactly. But other than that, they signal in the same way, bind to the same receptor that signals to the same, this G protein coupled GS pathway. So other than that, they work the same way as the endogenous hormone.
Mm-hmm. And so when GLP-1 or these GLP-1 drugs like Ozempic are working, when they're doing their thing, what are the effects on the GI system that are related to the satiety signal? So the brain is being signaled satiety, but what's happening to the GI tract? Is food moving through the GI tract less? Are other things being secreted, like other gut hormones? What is the GI system basically doing in response to this hormone?
Yeah, so one of the most well-described effects is a slowing of gastric emptying, and whether that contributes to the weight loss, I think is not 100% clear. But to my knowledge, some of the earlier versions that were less efficacious on body weight also had the same slowing of gastric emptying that maybe speaks against this as a major contributor to the weight loss. But in general, they are slowing gastric emptying. So basically food moves slower through the GI system. If the drugs affect secretion of other endocrine factors from the gut, to my knowledge, I don't know. I wouldn't think so. There's also in many hormone system, it's well known that if you kind of introduce some hormone replacement type of therapy, that you kind of shut down the endogenous production. And to my knowledge, that's not reported for the GLP-1 system, which is also quite fascinating. So then I don't believe you shut down the endogenous production with the drugs.
Okay, interesting. How well do these GLP-1 drugs work? Can you give us a sense for that in terms of the magnitude of the weight loss that people are seeing when it works, as well as the percentage of people that it actually works for?
Yes. So the first generation of these drugs were approved by the American authorities, the FDA, in 2014 and marketed under the name Saxenda. And this is the drug we just talked about called liraglutide. This was a once daily injectable, and it had approximately 8% weight loss over 1 year in individuals with a BMI above 30, so what we classified as individuals with obesity. And then there was this development with going from the liraglutide to the semaglutide with the once daily to once weekly. And I think it was a little bit, it was just I think it was unexpected that the weight loss was so much potentiated with that modification. So the once weekly drug in individuals with BMI above 30, 1 year trial, you see weight loss of 15% approximately, plus minus. There's now been so many trials, but up to 17% with this type of best-in-class GLP-1 receptor monoagonist. And typically then we talk about the Novo Nordisk compound semaglutide, which is kind of the leading one when we talk about the monoagonist drug. But there's also then the dual agonists that are now entering the market, and maybe we can also talk about them later.
Yeah, we'll get to that later. So the drugs that are out there, the once weekly injectables, the Novo Nordisk version, we're talking on the order of 15% loss of body weight.
On average in individuals with obesity, BMI above 30. And then out of those 15% average, there's of course a large variability in how people respond to these drugs. So typically around 15% we classify as non-responders, meaning they get less than 5% weight loss. But it also means that, of course, there are people in the other side that loses a lot of body weight, up to 30% weight loss on these drugs. And that's it's something we don't know why that is. People respond so differentially to these compounds. Why
is there a relationship between how obese they are and the magnitude of the effect? Do the most obese people tend to lose the most?
Yeah, good question. I believe so. So most of these trials are done in rather homogeneous populations. So typically in individuals with obesity. So I haven't seen any trials with individuals that are rather more lean. So that's more anecdotally, right? That you see people with less obesity respond less. And then there's also another kind of complexity is that people with comorbid obesity and type 2 diabetes, they also actually have less of a weight loss response. So there's something with the type 2 diabetes that weakens the effect on the weight loss that we also don't know why.
I see. So people with obesity and type 2 diabetes tend to lose less weight with these drugs than people who have obesity but not type 2 diabetes.
Exactly. So if the most beautiful data from the phase 3 trials is typically in obese patients with obesity without type 2 diabetes, because that's where you get most weight loss typically.
I see. And so when earlier you said about 15% of people, about 1 or 2 out of every 10 people that's obese that takes this drug is not going to see a significant response. It'll be 5% or less body weight loss. Does that include those people with type 2 diabetes potentially? Is that one of the things feeding into that 15% number?
They should probably not. I think that they've already been selected out of these type of trials, so it shouldn't be.
I see. So about 15% of non-diabetic obese people will not see a lot of weight loss from these drugs.
Yeah.
Okay, but still, that means a vast majority do see some, and many of those see quite substantial weight loss.
Yeah, and that's also why we see they're so popular, right? So I think finally, probably it's the first safe and efficacious weight loss that has entered the market. I mean, up until now, right? And this is also why they've been so popularized, probably.
Yes. And I want to come back to the safety question in a minute. First, I want to ask you about the weight that they're losing. So for people that lose weight on Ozempic and similar drugs, what are they losing? Is it primarily fat? Is it primarily muscle? Is it a mixture of both?
Yeah, so I was actually just trying to look up on these data because in many of the clinical trials, they haven't done body composition on all of the patients, which I think is also it's just too difficult of a measure as compared to body weight to do. But it's been done on subset of patients and in some of the follow-up trials. So typically, its data suggests that 25 to 40% of the weight loss stems from loss of lean mass or of fat-free mass, so to say. And then again, how much of the fat-free mass is muscle mass is then probably 50%, and the rest is comprised of the rest of the organs, right? But if we just talk about fat-free mass, because if you do DEXA scanning, that cannot differentiate necessarily between muscle and, so it would just say, let's just say fat-free mass, non-fat mass. And then up to 40% of the lost weight in some of the studies with the GLP-1 agonist is coming from fat-free mass. And historically, if we look at dietary or diet restriction, I think typically the number is around 25% with kind of normal low-calorie weight loss type of studies.
I see. So with these drugs, you're seeing a higher loss of fat-free mass than you would with just a raw food restriction. Is that what you're
I
saying?
think we don't have the data yet, and at least so I've seen ranges from 25 to 40%, and I also looked at Eli Lilly's drug, which is we can talk about this dual GLP-1 GIP agonist. It seems as if it's also around 25% in that one study that they reported. So I think the jury is still out, but of course it's a very hot topic. It's something that is being discussed widely now.
Okay.
Is this kind of risk with the loss of muscle mass?
Yes. So regardless of what, the numbers actually are, there's some loss of fat-free mass.
Yeah.
Um, is that okay? Is that not ideal? Is that dangerous? How should people think about that? Should they want to preserve that fat-free mass, or is it okay to lose it?
So I think there's two ways to view this. First of all, this will be very individual, right? Both depending on your sex or gender and your age and starting point. So I think if you're old and somewhat frail, you want to, at any cost, you want to preserve your lean mass, because we know that's directly correlated with how long and how well you live, right? You want to be able to walk up the stairs and grocery shopping, etc. So I think once you've acquired fat-free mass at old age, you want to preserve it at any cost. But for some individuals, I think it's expected. We know if you have a higher body weight, with elevated body fat mass also comes elevated fat-free mass. So typically individuals with obesity will have more fat-free mass also compared to, yeah.
So if you go from being lean to being obese, you're not just purely gaining fat mass. You're gaining some amount of everything. It might disproportionately be fat mass, but you're also going to be gaining fat-free mass.
Exactly. So that's also what if we kind of acknowledge this 25% loss of fat-free mass from the diet restriction studies, maybe that's expected, right? But maybe we want to make sure that with the drugs, we don't accept or we don't go beyond, for example, those 25%. Maybe that's a target to begin with, right? But that being said, why not preserve the lean mass we already have acquired? So I think that's the ideal future, right? That we come up with interventions or strategies that ensures that we can preserve as much as the lean mass that we've acquired as possible and exclusively lose body fat mass. And this is, of course, something that the pharma industry, but also academics are working on right now to come up with ways to ensure we can preserve lean mass. They can be lifestyle interventions, or they can be kind of drug co-administration type of interventions.
And when we look at the clinical data in humans in conjunction with the preclinical data that would have been done to justify those clinical studies, and we look at the weight loss, how much of that weight loss can be explained by a reduction in food intake? Or in other words, is there also a change in things like basal metabolic rate that feed into this?
So this is also a frequently debated question. So the rodent data is less clear. Some of the rodent studies suggest that there is an effect on energy expenditure with these incretin hormones, that you see increased thermogenesis. But the human data suggests that it's exclusively driven by a reduction in food intake. Maybe there is an effect on preservation of energy expenditure. So we see with a severe calorie restriction, if we go on a diet, we see that energy expenditure drops in order to kind of protect body fat mass. So that's what contributes to prevent us from successful weight loss, is a decline in energy expenditure. Maybe the drugs can alleviate that a little bit.
I see. So sort of the natural condition is that when an animal is faced with starvation or calorie restriction conditions, there's less food coming in to supply and to fuel the body. And so there's a compensatory drop in baseline metabolism so that the animal's using up what it has less.
Exactly. So we see a quite pronounced metabolic adaptation in order to protect its body weight and fat mass. So that's just well described both in humans and in animals. And then some of the drugs can maybe offset that reduction in energy expenditure slightly. That's at least a hypothesis. But a direct effect on energy expenditure, I think in humans is probably not one of the mechanisms.
Okay. So the weight loss that many people see is largely coming from a decrease in food intake.
Exactly. Yeah.
And so when people take these drugs subjectively, what do they report? Are they simply less interested in eating? Do they feel full sort of more of the time? Do they feel sick or nauseous or anything like that? What is the subjective feeling that these drugs induce?
Combination of all these things you just listed, right? So it's less interest in food, kind of inability to finish portions they've kind of piled up on their plate. Um, some background nausea for some individuals. That's also one of the main side effects is nausea and vomiting, right? So I've heard one individual, an older woman, say that it kind of she had the sensation as she recalled from being pregnant 20 or 30 years ago, like this background nausea all the time, and that's exaggerated when she was eating. So that's why you want to eat less if you so that so it's a variety of these kind of experiences that we hear from individuals taking the drugs, but in general, just less drive towards eating, basically.
So with that in mind, so there are at least anecdotal reports that people are quitting drinking, quitting smoking. They're having help with substance abuse that they might have, and that sounds like a good thing. But I want to, think about that through the lens of another weight loss drug that we saw come and go historically, and that is rimonabant. Do you remember the case of rimonabant? Can you give people just a very short summary of what that was and what happened there?
Yeah, so rimonabant was a very good idea because it was kind of blocking this cannabinoid receptor that we know is related to, or for most people, it's something they relate to kind of consumption of marijuana or similar products that contains this THC that will elicit this receptor in the brain that we know kind of promotes hedonic food consumption, so overeating basically. So this kind of links with marijuana consumption and overeating was a good idea to come up with that. Why don't we block this receptor, this cannabinoid receptor in the brain, and then we can suppress satiety, or we can increase satiety, but maybe even suppress kind of this hedonic drive to eat palatable foods. And this was developed, this drug, cannabinoid, I think 1 receptor inhibitor, and it worked quite well, but it also had a lot of, as it turned out, quite severe adverse effects, probably because it was targeting systems deeper in the brain than what we normally refer to as these kind of homeostatic systems. We talked about the circumventricular sites. I believe rimonabant might have been reaching deeper into the brain and then playing around with other brain systems, right?
I see. So the problem with rimonabant is that although it did help people lose weight, it was basically making them there were psychiatric side effects. People were feeling depressed. There was even suicidal ideation. So people were less interested in the rewarding properties of food, but they also became less interested in the rewarding properties of basically life itself. Everything was less rewarding, and the problems associated with that were greater than the benefits from the weight loss side of it, and so that was pulled from the market. Now, the fear would be that with the Ozempics and the new weight loss drugs, we're just going to sort of replay that episode. And so one way to interpret the anecdotal reports of substance use, people quitting substance use of alcohol and other things, is that, well, we're just seeing another rimonabant. It's tapping into the reward system, generally speaking. And so food is less rewarding and people are eating less, which is good because that's what we want here. But it's also making drugs and alcohol less rewarding, and by extension, perhaps other things which could lead to bad outcomes. Do you think that's likely, or it sounds like maybe you think that's less likely because the drug is sort of restricted to these circumventricular organs and it doesn't get into these deeper parts of the mesolimbic reward system and other places?
I mean, we know it influences dopaminergic signaling from rodent studies. So we actually coincidentally published a study last year in Cell Reports where we showed that we were interested in the interplay between nicotine and the GLP-1 receptor system. And then we teamed up with a group of researchers that had, again, this photophotometry setup where they could record dopaminergic signaling in the nucleus accumbens of the reward pathway. So every time the dopamine receptor is being activated in the reward pathway, we will get like a response in a recording. So if you give the animals nicotine, we see that this dopaminergic pathway is lighting up basically. And then we co-administer it with a GLP-1 receptor agonist. And we saw that the GLP-1 receptor in itself didn't do anything to dopamine signal. But once we gave it together with nicotine, we suppressed the nicotine-induced dopamine signal quite potently. And this must be indirect. So again, this is how is it that probably through the brainstem circuits, how is it these circuits are tapping into the dopaminergic system to kind of fine-tune it in a way that then suppresses the dopaminergic response we get to nicotine. And I think I think this is some of the mechanistic underpinnings of the potential effects of these drugs on substance abuse, right? That they are kind of playing with the dopaminergic system, but maybe we're fortunate in this case that it's the indirect effects that renders it quote unquote safer than what we saw with rimonabant. I think one thing we already know is that so many thousands and thousands and thousands of large trials have been done. There's now the longest one where patients have been 4 years on treatment. And to my knowledge, there's not a very frequent reported side effect, any of these kind of anhedonia, these type of kind of more kind of depressive type of states, for example. So I don't know, but it seems
Well, it sounds like a pretty informative experiment to do in animals would be to repeat what you guys did with the GLP-1 plus nicotine experiment, but for a reward, a healthy reward, something that we don't want people to get sick of and to quit, like, exposure to conspecifics, exposure to littermates, exposure to water. I don't know, something that the animal likes that is healthy for it, rather than something that it likes that it gets addicted to.
Yeah, so I actually had a journalist calling me up last week, said that she's talked to this kind of a very, very kind of not the, what do you call, not kind of a little bit of a tabloid type of press that said she talked to a woman that reported that she lost her sex drive with these new kinds of drugs. And she asked me, is there any kind of scientific foundation to support this? And again, I could speculate like we do now, is that, well, we know that probably the sex is one out of several natural rewards, right? So I think we can envision a way in which these drugs could influence these type of natural rewards as well. But it's, at least again to my knowledge, not a very common side effect reported. So it may be for some individuals, right? But we'll have to see.
Yes. So the next area I want to ask about these drugs is long-term effects and side effects. How long lasting are the primary effects on food intake? Are there any side effects? And sort of wrapped around this whole question is, how long have people actually looked? Has there been enough time to truly know what the long-term effects are?
So as I just mentioned, I think there was just 2 weeks ago published a study on semaglutide with 4 years on treatment, where they showed, I think I was a little bit surprised to see they actually show no weight rebound, because I think even for the bariatric surgeries that works extremely well, you see eventually some weight rebound over years, right? But at least on the 4 years on treatment, that they saw preserved weight loss, stable weight loss throughout the years. It was only, I was a little bit surprised, it was only at 10% in this study. So again, it's not the magnitude that we oftentimes report or want to, right? But still, it was quite interesting. And then regarding the long-term adverse events, for some of them we will have to see, right? But I think you can also flip it around, right? So the reason why the stock for these drug companies, most notably Novo Nordisk and Eli Lilly, is going up these days, is that they continue to come up with reports showing benefits on cardiovascular disease, benefits on kidney disease, benefits on osteoarthritis, benefits on whatnot. They're now doing phase 3 studies with Alzheimer's disease. So I think it's more the opposite, right? That they continue to show benefits on actually endpoints that are very important to human health. So I think one could also envision a future where we see next generations, like imagine this is your kind of iPhone 3, right? Imagine once we've developed these compounds even more, like in 20 years, you can envision a future where everyone are taking these drugs because they are directly linked to human health span. So and I'm not a proponent, or I'm not selling the drugs right now, but I think if we continue to see benefits with no surprise adverse effects coming up with usage, right, I think we'll see, maybe it's the other direction, right, that we'll see more and more people want to take these type of drugs, and it probably not Ozempic or Wegovy or Mounjaro, what they call. We'll see next generation compounds being developed.
You recently did a study where you basically created a new version of these drugs. And can you set up that study for us? What was the rationale for this, and how exactly did that drug work, and why did you even think to look at this?
Yeah, so I'll try and do it somewhat short. So we became interested in the glutamatergic NMDA receptor. That is typically something you associate with memory research, cognition, and kind of this long-term potentiation. That's something with synaptic neuroplasticity. The reason why we became interested in the first place was that there was these studies with ketamine showing effects in treatment-resistant depression 20 years ago, and then there's been more and more research uncovering the mechanism for this. Why is it that ketamine seems to have long-lasting benefits in depression? And then some of the data coming out on the molecular mechanisms pointed to a role for BDNF, the brain-derived neurotrophic factor, some of the mTOR signaling pathways, and a lot of these pathways we're quite interested in appetite regulation as well. So BDNF is a very common pop-up in all the genetic studies of body weight variability, and if you give BDNF into the ventricles of the brains of mice, they'll lose weight also. So the link between ketamine, which is an NMDA receptor blocker, its benefits on synaptic plasticity and depression, and its mechanisms involving these pathways that we know is also interesting in satiety control, was the first kind of stimulate the first ideas to think about the uses of these type of channel modulators for obesity. And then we could see in the literature that there were already sporadic studies, actually a few human studies testing NMDA receptor antagonism for binge eating disorder, and we found studies in rats and monkeys also suggesting that it could have effects on hedonic food intake, but there's not many of them. And then at the same time, the human genetic studies of body weight variability clearly point to a role for neuroplasticity and glutamatergic signaling also. So these different pieces of evidence increased our interest in NMDA receptor channel blocking for weight management. So we started to do experiments with commercially available compounds, including ketamine and the approved Alzheimer's drug called Memantine, and then a drug that's not on the market called MK-801, but it's a very potent channel blocker. So we exposed obese animals for these drugs and looked at food intake and body weight, and saw consistently that they actually lowered food intake and body weight when we gave these drugs systemically.
So NMDA receptor antagonists, drugs that block this receptor that's important for plasticity and learning and memory, they tend to help with obesity. Animals tend to eat less and lose weight.
Yes, but it's not very consolidated evidence that you find in the obesity literature. It's kind of, you have to search for it to see.
I see.
Previous. So there's like a handful of previous studies that are spread across decades of work, basically.
I see. So we had some indications that NMDA receptors blocking them might help in obesity. Obviously, these GLP-1 drugs, you're very familiar with them yourself. They're out there, they're working in humans, approved, causing weight loss in some people. So what was this new drug? There's like a twist on this drug, right? You did something kind of clever to make it work in an interesting way compared to the other GLP-1 drugs.
Yeah, so the NMDA receptor antagonists are small molecules. So like rimonabant, they will basically penetrate the blood-brain barrier and signal throughout the brain. So we saw when we gave high doses of these drugs, the animal also started to move more. They became hyperlocomotive. Their body temperature increased. We saw a lot of adverse effects that it's just not a useful drug for obesity treatment. You cannot have a weight loss drug that has these adverse effects. But we thought we want to just exclusively harvest the benefits of these drugs in these circumventricular sites. So how do we ensure that they don't penetrate deeper into the brain? And this is when we start to make these peptide drug conjugates. So we took the small molecule and then through a chemical linker, we attached it to the peptide. So we basically created a single molecule with the small NMDA receptor channel blocker attached to the GLP-1 peptide. And then through the GLP-1 peptide not being able to penetrate deeper into the brain, but homing into its GLP-1 receptor. And when the peptide interacts with its G-protein coupled receptor, it is being internalized as a complex. And then you can sneak in the small molecule into those same neurons that internalizes the GLP-1 receptor, and suddenly you have cell-specific delivery of that small molecule only in GLP-1 receptor expressing cells.
Okay, so you've essentially taken the GLP-1 peptide and you've stapled it to an NMDA receptor blocker. And you've done that in a way that it can't get through the blood-brain barrier. So this drug can't penetrate into all of the brain, but it can get into parts of the brain, like the hypothalamus and the brainstem, that are directly touching the bloodstream, that don't have a blood-brain barrier there protecting them. And so it, because it's GLP-1 connected to this small molecule drug, it binds to the GLP-1 receptor on the neurons that have it, like the satiety neurons, the POMC neurons we talked about before, and then it goes inside the cell, and then the small molecule drug is cut off of that, and then it blocks the NMDA receptor.
Exactly. So, and I'm not a chemist, but fortunately, the first author on our paper and has been working on the project for a long time is a very smart chemist. So there's, from the antibody drug conjugate space in cancer treatment, there's been developed a lot of different linker chemistries in order to kind of facilitate delivery of a toxic agent only to cancer cells. So you want only delivery of kind of this payload to specific cells. So they've been developed a linker type of chemistry that is sensitive to the intracellular environment. So it will only be cleaved once it's inside the cell. So it can be, for example, pH sensitive or sensitive to other intracellular aspects that will ensure the cleavage is only happening intracellularly, but it stays stable in circulation.
I see. So from a chemistry perspective, this thing can be engineered so that the sort of double molecule, the peptide plus the drug, can't be broken. It's stable unless it goes inside of a cell, and it can only get inside of the GLP-1 receptor expressing cells.
Exactly. Yeah. So that is the idea. And then you have liberation of the small molecule within those cells, and then you need them to block the NMDA receptor in those same cells.
Okay, so sort of the end effect physiologically of this novel compound is that you're able to block NMDA receptors in GLP-1 expressing cells only.
Exactly. So it's a very, very specific delivery of that small molecule. But we believe the idea is that is perhaps the cell type we want the signaling of those molecules the most anyway. So why deliver them through the whole brain when we can get them only to those specific neurons?
Okay. So my next question would be, what is the neural, so of the neurons that have the GLP-1 receptor that are sensitive to this drug, what is the effect on neural activity that this drug has? Because on the one hand, we said before that GLP-1 renders neurons more sensitive to their inputs, so it should make them more excitable. But the NMDA receptor blocker is going to prevent NMDA receptors from opening and depolarizing the cell. So what's the net effect here?
Yeah, so temporarily, so the idea is that the NMDA receptor is in interaction with the AMPA receptor being recruited. Once the glutamatergic drive is sufficiently high, you get NMDA receptor, you remove this block by magnesium, and you get signaling through the NMDA receptor. So the idea is, I think in terms of plasticity, that continuous blockage of the receptor might increase the glutamate in the synaptic cleft, and then subsequently it might elicit some of these adaptive responses to strengthen the neuronal connectivity. But all of these details, the short answer is we don't know what is happening for all these details at this point. But some of the things we did was that we teamed up with a group of researchers at the University of Texas that is doing slice electrophysiology. So they take out slices of the hypothalamus and they can bathe them in this kind of specific buffer, and then they can patch onto individual neurons. And then they have labeled the GLP-1 receptor positive neurons. So these mice come with a labeling of a neuron. So they know they're recording from a GLP-1 neuron in this artificial setup. And then once they put NMDA or glutamate on these neurons, you see a depolarization and an inward current. And then if you put these channel blockers on that have been developed to block the channel, you see that this blockage is you prevent completely this inward flux of ions. And if you put GLP-1 on, nothing happens. You see again this NMDA-induced inward current. But with the conjugate, we see that we can mimic what we see with the small molecule. So this is one way of showing that we have retained the effect of the channel blocker in this type of ex vivo setup. In addition to those type of experiments, we also we took out hypothalami and brainstem from mice that have been treated with the drugs. And then we use these modern omics technologies where we look at all the transcripts and proteins that are changed in these brains of these animals. So all the mRNAs and all the proteins. And then we see that with the conjugate, we see ton of pathways related to neuroplasticity, glutamatergic signaling. So proteins are being upregulated that is involved in synaptic plasticity. It's an indirect measure, but still it's a quite clear signature that we see changes in synaptic plasticity with the conjugate. Mm-hmm. So we did these different types of experiments to strengthen that the concept works as we have hypothesized, right? But we're still doing this type of studies, setting up studies now to verify this specific neuronal modulation in vivo through both the GLP-1 receptor and the NMDA receptor.
All right. So then in essence, a lot of the experiments you did were in rodents. You're comparing NMDA receptor blockers like MK801 to semaglutide, the GLP-1 drugs. So the normal GLP-1 drugs that are out there on the market already, NMDA receptor blockers, you can compare those two. And then you can compare both of those conditions to the new dual action GLP-1 plus NMDA receptor blocker drug that you just described.
Yeah.
How do in terms of the basic effects on weight loss and metabolism, how do these drugs compare in your hands?
Yeah. So this is normally we say the predictability or translatability of drugs, weight loss drugs in rodents is quite good. They translate quite well to humans, but there's one caveat, and that is once the drugs become very potent, such as the semaglutide or some of the Eli Lilly's tirzepatide, etc., in rodents they're so potent that they can basically clear the animal for excess body fat. But we can do studies where we can kind of tease apart what is the contribution from the NMDA receptor component. And that's, of course, what we're interested in, to show that there is room for maximizing the weight loss benefits. So one study we did is that we pre-treated obese animals with semaglutide, or Ozempic basically for 2 weeks once they start to plateau. Then we randomized them to stay on semaglutide plus additional GLP-1 on top, or we added our conjugate on top of semaglutide. And then we saw we could push body weight down 8% or so further, again, suggesting that additional weight loss, when you have maxed out on GLP-1, that we can get additional 8% or so on top with the NMDA receptor channel blocker. So those type of experiments we tried to do in order to show how much we could gain from the channel blocker.
I see. So you actually gave rodents semaglutide, the GLP-1 drug, and additionally at the same time gave them this new dual action drug, which is also a GLP-1 drug plus the NMDA receptor blocker. And you're saying that you saw additional weight loss. So you sort of maxed out what you can do through GLP-1 by itself. And then on top of that, if you add the NMDA receptor blocker in specific cells, you're getting an additional, quite substantial amount of weight loss.
Exactly. So those type of studies we did, and we did direct comparisons where we infused the molecule directly into the brains of rodents, so in the ventricular system, and a molar matched dose to semaglutide, single infusion, and we saw even I think a doubling of the initial weight loss compared to semaglutide directly in the brain. We saw a little bit more preserved or slower rebound also compared to semaglutide in that specific study. So But because our drug in the paper we reported doesn't have this lipidation, the fatty acid side chain, it's difficult to directly compare to the marketed drugs.
I see. But in these experiments, when you compare semaglutide, the GLP-1 drug, to the new dual action drug, not giving them at the same time, but giving them separately to different cohorts of mice, the new drug is causing more weight loss.
So that is, so I think that is the point I was trying to make before. So there is a handful of drugs now that if you dose them high enough in rodents, they will basically plateau at the same point, right? So you can clear their body fat. So that's where you have to come up with, you have to either dose a little bit lower to see how they differentiate, or you have to kind of do this as we did on top to see if there's additional benefits that can be harvested.
But I guess the point of these experiments is there is additional fat mass that can be lost through adding the dual action of this drug, the NMDA receptor component, on top of the GLP-1 component.
Exactly, yeah.
What about other effects, physiological effects that are relevant to metabolic health? I know that you guys looked at things like glucose homeostasis and insulin. You looked at other things. What are some of the other effects that you saw with this drug?
So importantly, we saw across the board, like markers for metabolic health were improved, like circulating markers for lipid metabolism, lowering of circulating cholesterol, triglycerides, and improvement in also glucose metabolism. So those are the things you need to see nowadays with a new drug, right? You need to show benefits on several metabolic parameters. Then we showed, at least in rodents, again, we didn't see any adverse effects on blood pressure or heart rate, which I think is important, again, to demonstrate you don't have any cardiovascular adverse effects. And then we saw some tendency also to this effect on energy expenditure. I think that was quite interesting. So we actually saw quite a substantial protection of this metabolic adaptation. So even though the animals in this experiment has lost 30% of their body weight, the energy expenditure was as high as the control animals that were still obese.
I see. So that's interesting. So earlier we talked about this. When animals start eating less, there tends to be a compensatory decrease in their energy expenditure. You're saying that that's absent with this drug. So animals are eating less, but their energy expenditure is staying where it started at.
Exactly. And in this study, we had a control group that we calorie restricted to match the weight loss of the drug, and their metabolism was substantially slower. So they just basically decreased their energy metabolism dramatically. And we didn't see that with the treatment. They completely retained the energy expenditure throughout, despite losing 30% body weight. So that could be one important aspect. Same goes for actually lean mass. We do see a little bit of lean mass loss, but again, compared to calorie restriction, we actually see a preservation of lean mass in the rodents again. So we don't see exaggerated lean mass loss either.
And because this new dual action drug has more specificity, I would imagine that inherently there's less chance or less likelihood of side effects because you're only affecting NMDA receptors and only in these neurons. Did you guys look at that at all? Like any negative side effects? Is that true? Is that like a likely way that it would work because it has that higher level of specificity?
So we did some of the most obvious tests for side effects that are very apparent when you use the NMDA receptor channel blocker on its own. So we used like this open field arena where you would just video record the animal after drug exposure, and then we dosed very high, so we could see that the MK801 treated group, they just ran around like crazy, hyperlocomotive. But in the conjugate, dosed equally high, we saw absolutely no effect on hyperlocomotion, again showing that we don't have this broad central delivery of the drug. We saw that we did the same for hyperthermia, like the elevated body temperature. We didn't see any adverse effects on body temperature either. So we did this kind of tip of the iceberg experiments to show that we don't have the adverse profile of the NMDA receptor channel blocker. So I think it's quite fair to say that we do have this specific delivery, and we don't have systemic liberation of the small molecule.
And what effects did you see in the brain? What's going on to neurons in the hypothalamus and elsewhere when you give this drug?
Yeah, so in the paper, we quote unquote only did these type of omics experiments where we looked at the whole transcriptome, all the mRNAs, all the proteins in the hypothalamus. We did in the brainstem, and we also looked in the nucleus accumbens, and then we did whole brain 3D imaging where we imaged a protein called CFOS, which is a indirect marker for neuronal activity. So when a neuron is activated, CFOS is being expressed. And then if you stain for CFOS, it will indicate that that region has been activated.
So basically, you can do an experiment where across the entire brain, you can look to see which neurons have recently been quite active.
Exactly. So the brain it's a very neat technique. The brain is completely made completely transparent, and then you can basically visualize 3D the whole brain where neurons have been activated. So it's a very fascinating method. And what we see is that we see activation of the same regions. We did the control against semaglutide and the monoagonist, etc. We see the same regions as we know are being activated, the brainstem, the hypothalamus, but we also actually saw some activity in the nucleus accumbens with the conjugate, which were quite interesting. Again, the molecule is unlikely to go there, so it must be some type of feed-forward signal from possibly brainstem that activates this region of the brain.
Okay, so you see neurons become quite active in the hypothalamus and the brainstem, as you would expect, because this is where the neurons live that we know are affected by these drugs. But you see additional neurons elsewhere in the brain, in particular the mesolimbic dopamine reward system, that also become active. Do you know the specific neurons that became active? Are these dopamine neurons themselves?
We don't know at this point. This is just only CFOS in regions that we know are quite well defined, right? So we have no idea what these specific neurons are. Not at this point.
So you sort of see the same areas lighting up as with semaglutide, but then you see these additional areas lighting up, and we don't really know much about what that means yet, it sounds like.
No. Yeah. And some of the areas that we see with semaglutide, we also see, and for example, an exaggerated response again, which perhaps can be explained by part of the mechanism is that somehow you potentiate the GLP-1 receptor signal with the channel blocker in those neurons. Again, this is speculative, right? But how is it working? It might be that we're kind of boosting the GLP-1 receptor signal in some way through the NMDA receptor channel blocking.
And are these drugs affecting other aspects of metabolism in any way, like lipid oxidation and sort of how the calories are being used?
I mean, they shouldn't. Yeah, I mean, they shouldn't be too much beyond what we see with the GLP-1 because we have this exclusive delivery, and the NMDA receptor is predominantly expressed in the central nervous system. There is an interesting paper published quite some years ago in Nature Medicine showing that NMDA receptors in the pancreatic beta cells is involved in insulin secretion. And we've had many questions presenting this at conferences where people have suggested us to look more into this. So it could actually be beneficial also for even further potentiating insulin secretion, at least based on the literature. But we haven't spent much time on this as we're mostly interested in the brain, but it's something that we should look more into.
So one of the things you mentioned about NMDA receptor antagonists is they have a number of side effects, including hyperlocomotion. But actually, for something like obesity, you could argue that that's an effect rather than a side effect. How are these drugs affecting baseline locomotor levels? Did you look at anything like voluntary wheel running or something that we might think of as akin to exercise in rodents?
Yeah, we actually did. So in this calorimetry system where we measure the animal's energy expenditure over 10 or 12 days of treatment, we can also pick up on locomotor behavior. And here we actually don't see any differences compared to vehicle-treated animals. But we see that animals that are exposed to calorie restriction, they reduce their locomotion, which is a way to preserve energy. In wheel running studies, we've established wheel running model as a kind of model for aversion in rodents. So we see when we give an aversive substance, the animals, they don't like to run in the wheels, so for us, we've tried to establish this as a model for predicting aversion or avoidance behavior. So they avoid to run in the wheel. So we compared the wheel running experiments next to semaglutide to see how this affected their willingness to run. And we saw a similar profile. The first time they see the drug, it is aversive as any other GLP-1. They don't like to run in the wheels, but on the subsequent days, they start running again. So I think the aversive effects are quite transient in the rodents.
I see. So there's an aversive. They stop doing things they normally like doing for a little bit, but then they get used to it basically, and they start doing them again.
We see this with all of these weight loss drugs that hits the brainstem, that they have an effect on the willingness of the animals to go in the running wheels, which is quite an interesting model, I think.
Interesting. Is anything known there about, like, in humans? Have they looked at, are there any reports about people's physical activity? So they're feeling less hungry, partially by just probably not feeling interested in food, partially through nausea and malaise. So it's kind of an aversive state maybe that's being induced. Are people reporting that they are exercising more or less or the same amount or anything like that?
Good question. I don't know. So I think in the clinical trials, there's always like this, the drug is always being prescribed on top of lifestyle therapy, like recommendation to eat healthy, live healthy. But I actually don't know what is being reported back, how much the subjects are increasing or decreasing activities. That's something that would be very interesting to look into in the future. Also, I think for some individuals, you can imagine that if they alleviate some of their issues with moving around, if you lose a lot of body weight, you are able to move more, like due to your physical size. But I don't know centrally if it's going to be affected by your kind of predisposition or willingness to move. That I don't know.
So given the experiments that you did, that you just described, where you've made the drug more specific by basically making it a dual action drug, it's affecting only GLP-1 receptor expressing cells, and it is then blocking NMDA receptors in the neurons that have GLP-1. It's, everything that in your paper sort of looks good. It sort of improves everything. There's more weight loss. Everything's as good or better than what you see with the GLP-1 only drugs.
Yeah.
There wasn't a lot of negativity to report in terms of how I read your paper. Do you think that these types of drugs will just sort of supplant the existing GLP-1 drugs? Where do you see things going in the next few years?
Yeah, I know that. So here we have to have a look at the current kind of landscape and pipeline that is going through the clinic, right? So already now we can say that the GLP-1 monoagonist semaglutide is being heavily challenged by the dual and triple agonists that are either on the market or coming on the market. So there is the GLP-1 GIP co-agonist developed and marketed by Eli Lilly. It's marketed as Mounjaro for diabetes and Zepbound for obesity. The clinical data shows that it has a few percent more weight loss benefits than GLP-1 alone. So this is the first kind of dual agonist that shows additional benefits. And then there's also a triple peptide agonist in clinical development, also from Eli Lilly, targeting glucagon. GLP, GIP also show very promising data. At the same time, the other companies are developing co-administration versions with a long-acting version of a hormone called amylin that have been developed also for weight loss. So you give a GLP-1 together with amylin. So there is like an endless number of combinatorial ways that are being attempted right now to potentiate weight loss to go from 15 to 25% basically. So that's ongoing. And next to that, there is also then the lean mass preservation initiatives where people start to tap into this myostatin system. So it's targeting skeletal muscle, inhibiting muscle atrophy pathways in order to preserve muscle mass while giving an appetite suppressant. So there's many different initiatives. And then coming back to our initiative is, of course, how is that going to fit into this competitive space? So we'll have to see, right? But we think that this the targeting the neuroplasticity could be might show some benefits that we don't see with the other drugs. And here we're dreaming about, our vision is to develop therapies that will have more sustained effects. So if you kind of rewire some of the appetite or hunger pathways in the brain, you may be able to actually create a drug with more sustained effects and less rebound. So again, it's the obesity drug market is increasingly competitive, but there's also room for many new type of therapies. But it's a very exciting field right now.
I see. So we've got these weight loss drugs that everybody's talking about that many people are using, but there's room to improve them in the sense of making them cause more weight loss, there's room to improve them in the sense of preserving the non-fat mass with the fat loss. There's sort of all of these different, you can make them more specific so they're less likely to have side effects.
Exactly.
And it remains to be determined what the long-term effects will be. But it sounds like you're pretty optimistic about these drugs.
I'm pretty optimistic right now. I think it's so far so good, right? But again, we'll have to see. But in parallel, there's been production issues. They're very costly to make these peptides. But right now, we also see development of small molecule GLP-1 receptor activators. So there's so many interesting things happening right now. So in order to get weight loss drugs to as many individuals as possible, you also have to be able to produce it easily and at low cost, right? So those are some of the issues that we don't talk about that often, but we also need to talk about, right? Because right now you're kind of catering to the part of the population that has a quite high income, right? Because they're pretty costly, but eventually they will also go off patent, and the competition will be bigger.
So insurance doesn't cover these drugs for everyone. Like, does it cover them if you're type 2 diabetic? Does it not cover, does insurance not pay for these drugs if you're merely obese?
It's very country specific, so I can it's tough for me to answer. I guess I should know for the US, but to my knowledge, it's very expensive to get in the US. And I don't know how much insurance is covering in Denmark right now. It's also somewhere between $300 to $400 a month to get the drugs, which is, again, depending on your income level, it's quite a bit of money, right? Especially if you're in the lower socioeconomic classes where obesity prevalence is highest.
Yes. What about do we know anything about, so people are losing weight because they're eating less when they take these drugs for the roughly 85% that will see that effect. Are they eating different foods? Are they eating better? Are they merely eating less of what they ate before? Is it having an effect on the palatability of food and what they have a taste for, and their propensity to eat healthy versus junk food?
So I think in the rodents, these type of studies have been done, and they show convincingly there's a change in macronutrient preference. In humans, again, I think the individual variability is quite vast, right? Again, anecdotally, you have reports saying that I never touch a dessert anymore, or I haven't drinking alcohol since I started on the drugs. I don't like coffee anymore. I used to buy a whole pizza. I still buy one, but I finish half of it. And so it seems to be, there's a huge variability, at least anecdotally, right? I'm not aware of any scientific.
What are the rodent results in terms of the macronutrient shift?
I believe so. I think Randy Seeley did one of the first studies, basically just showing that a change from less fat preference to more carbohydrate preference. Typically, I mean, it's a little bit of the setup has limitations, right? Because you give animals access to like a pretty boring versions of casein, protein, some lard, fat, and then some whatever carbohydrate source, and then you basically see how does it kind of prefer these three different macronutrients in an untreated condition, and then what happens to its preference once you treat them with a variety of drugs. So it's kind of a limited a buffet type of intervention, but I think as far as I recall, it changes preference from fat towards carbohydrate and keep protein at the same level.
I guess a little bit more of like a philosophical or societal level. If we just sort of extrapolate forward, where do you see things going in terms of the development of these types of drugs and therapies, and the way that sort of we interact with all of the things that we consume in our society? Like, 10, 20, 50 years from now, are we all going to be waking up and, taking a shot of the GLP-1 agonist in the morning and some testosterone? Are we just going to start injecting ourselves with, 5 different drugs that have been engineered with such specificity that we all just sort of create the body that we all desire without having to do anything else about it?
Yeah, I mean, I'm happy to try and speculate, but then again, if there's some adverse effects with this drug class being reported in 2 or 3 years from now, it can be completely scrapped, right? And we'll have to start from scratch. But let's imagine that we don't see this adverse effects with the incretin drug class, then I think, as I speculated earlier, I have a hard time envisioning a future where this is not a type of drug that many people will take as they age. Specifically, I mean, we'll see the data from the phase 3 trial on Alzheimer's disease in 25 or 26. If it has benefits on preventing the progression of neurodegenerative disease, if it has benefits on cardiovascular kidney disease, I mean, you can view it as kind of health span type of enhancing drug.
Yeah.
And again, it will not be semaglutide or Wegovy. It will be next iteration of these type of molecules that are being refined, and maybe again they'll be more individualized or personalized, catering to different needs. I think most important is that the biggest fear is that we create a large separation between socioeconomic classes, right? So you basically see the rich get healthier and the poor gets no access to these type of molecules, and you see even further separation in inequity and inequality.
Yeah, I mean, you can easily imagine that happening because, if these things are expensive and they work, then only certain people are going to be able to afford them every month. So one question I have too, so related to just the general topic of hormone replacement therapy, a lot of people do, you see more and more people using growth hormone or testosterone replacement therapy and things later in life. And we know that a lot of those hormones, generally go down with age. And so you're sort of just compensating for this age-related decline that we naturally see with a lot of these hormones. Yeah. What about GLP-1's natural time course? Do we know anything about how that evolves over the lifespan?
Good question. No, actually, I don't. I would be interested in seeing like meal responses in very old individuals to see if they have impaired GLP-1 secretion. I don't think so. I haven't heard about it before. So I actually don't think it's changing with age. I wouldn't think so, but it's a good question. But I think for the hormone replacement therapy with age, I think, and this is outside my field, but I'm always worried that if there's a meaning with the decline with age that pertains to the risk for developing cancer, basically, is that maybe as the risk for cellular kind of uncontrolled growth increases as we age, maybe you want less growth factors to promote that risk. I don't know, it's speculation, right? So I think that could be one risk. But as we again become better with medicinal chemistry and targeted delivery, I mean, again, testosterone is a steroid hormone, and if we can think about ways to deliver it using peptides to tissues where we actually want it, and then prevent it from going to tissues where we don't want testosterone signaling, I think those are more the strategies that I'm interested in, right? So one can easily envision a muscle-targeted delivery of, let's say, testosterone, where you're probably not too worried about having that delivered, but you don't want it to the cardiomyocytes, to the heart muscle, right? Yes, so there's these things you need to figure out how to do.
Interesting. Well, so here's what I hope will be a fun but instructive question. Let's just say you wake up tomorrow. You look like you're a lean, fit guy, but let's say you wake up tomorrow and you've got a BMI of 30. So now Christoffer is obese. You're not diabetic, but you are obese. Walk us through the thought process that you would have for, thinking about, should you take one of these drugs? Which one should you take? How would you think about it if you were obese?
I think I would go ahead and start with testing. Either there's two on the market. There is it's Ozempic is marketed as Wegovy. It's semaglutide, and the dual GLP-1 GIP co-agonists from Eli Lilly is marketed as Zepbound. So those are the two drugs that are available from this class right now. I guess I think I wouldn't hesitate with trying with starting on one of these therapies if I have failed in all other attempts, as most individuals with obesity have for decades, right? They've tried all the diets out there and continue to experience failure upon failure, and you have the stigmatization. Why not for once kind of get help with the pharmacotherapy if that's now available, and if you respond well to the drug, you don't get the. I'll be worried about the adverse response in terms of nausea and vomiting, and then I would hope I would respond well, right? So I think those are my kind of thinking about these considerations. But
and with respect to those drugs that are on the market right now, is this a lifelong commitment? Do you need to stay on these drugs, or what do we know about weight regain coming off the drugs?
Yeah, so the few studies that have been done on this are very clear. Is the moment you stop treatment, the patients will regain weight, as we know from all other interventions that lower body weight, right?
And when they regain the weight, is it the same distribution of fat and fat-free mass as before?
That has yet to be determined, and again, that would be very individual. So you can envision a scenario where you've lost whatever kind of proportion of fat and lean mass, and then you can envision two individuals. One is going off the therapy and staying on the couch, and the other one is being very active. So as you are in positive energy balance, anabolic state, and if you at the same time stimulate muscle protein synthesis, you'll have some of the excess energy go into muscle building, basically. And if you have absolutely no muscle protein synthesis going on, you can imagine that most of the excess energy will be going into your fat depots. So I think the devil is in the detail with respect to how the weight regain will affect body composition.
So with respect to your lab and the research you guys are doing, what are some of the next steps for you guys in terms of these weight loss drugs? What are you working on now, and what's on the horizon?
So we're continuing. We're excited about this peptide-directed targeting of small molecules. We're excited about neuroplasticity in obesity, very excited. So we continue to think about other ways of targeting neuroplasticity for sustained weight loss. That's kind of our long-term kind of dream project, to be able to better understand weight homeostasis, to create a therapy that will have sustained effects with very infrequent treatments. And at the same time, we continue also to add on modes of action. So we have, of course, started to develop this dual incretin with the small molecule. So we have kind of triple acting peptide small molecules. And then we're starting to look at other indications also, especially neurodegenerative disease. So if the incretin hormones turns out to have a benefit on preventing progression of Alzheimer's disease, maybe attaching some of these NMDA receptor modulators to the incretins will actually even benefit those type of indications more. So we're looking at other indications also right now. So many different directions, but we're excited about the targeted delivery, neuroplasticity, and other indications for these molecules.
All right. Well, Professor Christoffer Clemmensen, thank you for your time. Fascinating stuff. I was happy that you decided to come back on and talk about some of the newest work. And I look forward to seeing what you come out with next.
Thank you. And thank you for the invitation. It was a pleasure.